Welcome to Educator.com. This is the lesson on the scientific method. When we look at the origins of the scientific method and how it exists in today's society, it's not just from the Western world. It didn't just originate in Europe hundreds of years ago. You tend to hear that a lot because, especially if you're taking biology classes or other science classes in the United States or in the westernized world, you're going to hear about what a bunch of dead white people did from hundreds of years ago. But it's not just from that lineage. There's plenty of evidence that the scientific method came up independently in all different parts of the world. For instance, in ancient Muslim philosophy, there's plenty of evidence that they had their own scientific method, a way of finding objective evidence in terms of how things work in their world. And that's really the whole point to the scientific method, is trying to find empirical data that is not influenced by subjectivity. It's getting to the facts and being able to replicate that particular experiment repeatedly with different people. Biological study tends to trace the roots back to the work of Sir Francis Bacon and René Descartes in terms of post-Dark Ages in Europe, getting into the scientific revolution, and these particular gentlemen having a lot to do with influencing the modern day steps in terms of doing the scientific method effectively and the right way. It attempts to allow understanding of phenomena in nature by obtaining clear empirical evidence. It involves a hypothesis, and this is not just a random guess. This is an educated guess. It's based on what you've observed already, and it's really using your experience and being very clear about, hey, I've observed A, B, and C, so I'm pretty sure that D will happen. So it's an educated guess. You obtain data in the experiment. It could be quantitative meaning having to do with numbers, or it could be qualitative. Ideally, you want to go quantitative. You want to have good numbers gathered into your experiment, measuring data with numbers, and that way any other person could measure the same numbers, and it's not really involved with as much subjectivity or opinion. But sometimes quantitative data isn't possible based on whatever particular question you're asking, so qualitative sometimes does work. If you want to see how someone's mood is affected by a certain food or a certain drug, qualitative data may actually be more meaningful then, because it has to do with observing the person and recording what you notice about their mood in a qualitative way, the quality of that person. But yeah, both of these are reliable in different circumstances. And of course, the forming of conclusions. You want to get an answer. You want to figure out, hey, what did the data tell us? What does that tell us about our question? And over time, you can get better and better answers as you come up with better questions, getting to the heart of the matter. With the steps of the scientific method, depending on what source you look in, you're going to see sometimes it's slightly different order or different wording in terms of how you go about doing this. But this is a classic way to look at it. First, you got to observe something in your environment that piques your interest. That's interesting that happened. I just observed that. So let's take a circumstance that's not in a lab necessarily. Let's say you're in your house or apartment. It's 9 p.m. at night. All the power goes out. You don't got any candles to light, so you go to the drawer where you keep your flashlight. Okay, so you take out your flashlight, you try to turn it on, it's not working. Darn it. So you just observed flashlight not working. You ask yourself a question. This might seem obvious, but the question is, why isn't my flashlight working? Well, your hypothesis might be, an educated guess, that, hey, the batteries are dead. The battery's no longer functioning. That's why this thing won't turn on. And that could be. It's not necessarily the answer, but that's a good educated guess. That is your hypothesis regarding this issue. And how are you going to obtain data? So data is just a fancy scientific word for information. And actually, most people don't realize that data is plural. A datum, D-A-T-U-M, is the singular version of that word. This is when you're actually experimenting. So a way that you could obtain data about this particular battery issue, you take out the old batteries, and hopefully in the dark you can find where you keep the new batteries. You put the new batteries in, you click the on button, well, either it's going to work or it's not. So that's where you interpret your data. You interpret your data, like let's say the batteries, you put the new batteries in and it still doesn't turn on. Well, you probably just proved your hypothesis false, that it's not the batteries. Maybe it's the bulb. Maybe it's the fuse, some other electrical wiring in the flashlight. So, you know, it really, depending on what your results are, that influences how you're going to continue. If in this particular flashlight case you realize, hey, it was the batteries because I put new batteries in, it's on. End of the story. Problem solved. Your conclusion is, hey, it was the batteries, I fixed that problem, moving on. But, you know, if it wasn't the batteries, maybe you have to go back and redo the experiment by manipulating something else. So it's all a learning process. If you know a lot about cars and your car dies and ends up on the side of the road and you want to figure out what's going on with my car, people use this without even realizing it. You want to test all these different factors and see how they're affecting the whole system. And it's all about isolating the variables and doing it in a very objective sense. So when we look at the scientific method in action, different groups need to be established. So let's say you have a goldfish at home, okay? And your goldfish is in its bowl. Let me do it in black here. And you can color it in yellow. There's the little goldfish. And the goldfish, you know, you're on your, let's say your 5th goldfish. There's the water it's in. And you're like, hey, you know, every goldfish I have only lives a few months, if that. I want to know, how can I prolong the life of the fish? Maybe it's the water. Maybe just tap water is not the way to go. Maybe it's something about the pH of the water, how acidic or how basic the water is. Maybe that could affect the life expectancy of the fish. So a control group is an attempt to mimic the natural environment. You want to get as close as possible to what the natural circumstances and variables are in that particular organism's existence in nature. So with this fish, it definitely has a native population that these goldfish have been taken out of, you know, to give to people as pets. So if you could figure out, hey, you know, in this particular population of goldfish in the wild, it's fresh water and the pH is pretty close to a 7. So maybe the control group is the pH of the water is 7. So you have your fish, a bunch of fish, ideally not just one. The more numbers you have, the more reliable your results generally are going to be. So you have a bunch of goldfish in water with a pH of 7, which means it's not acidic, it's not basic, it's neutral, just down the middle with pure water. But the experimental groups is where you actually manipulate the pH. You then change the pH in these other groups. So you'll have some fish in, instead of 7.0, you'll have some fish in 7.2, some fish in 7.4, some fish in 7.6. You want to go the other way too. You're going to have some fish in 6.8, 6.6, 6.4, etc. And, you know, the more groups you can have, the more you can hit those different pHs. Probably the more reliable your results are going to be in terms of the specificity of your answer. The reason why I only varied this by 0.2 increments is because pH is a logarithmic. When you go all the way to an 8 from a 7, an 8 is 10 times more basic than a 7. A 9 is 100 times more basic than a 7. So going from 7 to 8 to 9 to 10, you're probably going to have a lot of dead fish if you do that. It's the same reason with on the side that's lower than a 7. You're in the acidity range there. When you go from a 7 to a 5 is 100 times more acidic than a 7. A 4 is 1,000 times more acidic than a 7. Just to give you an idea of how this impacts us, with the pH of blood in humans, it's around a 7.4. So our blood is slightly acidic. But back to our goldfish example, you want to have those experimental groups where just the pH is changed, nothing else. Speaking of just the pH being changed, when we look at the variables, the independent versus dependent variables, this is how you actually would obtain your data by looking at these particular parts of the experiment. And if you wanted to display it graphically, this would have a big impact on that. The independent variable has to do with the question you're asking. So the question we're asking here is, what is the ideal pH for my goldfish in terms of prolonging its life expectancy? Well, the independent variable is the pH. That's what our question had to do with. That's what we want to manipulate. So in those experimental groups, the only thing that should be changing in those fish bowls or fish tanks should be the exact independent variable in terms of the quantitative value. The temperature of the water should be the same. The size of the bowls or the tank should be the same. You don't want one of the tanks to be a lot bigger because perhaps that fish being able to move around a lot more compared to its mates in the other particular tanks. Maybe that one gets a little bit more exercise and that influenced its life expectancy. So you want to keep all the other factors constant. I actually made a note of that down here. Other factors remain constant. The dependent variable literally depends on the independent variable. So that's how I keep it straight in my head. The dependent variable depends on the independent. And really what depends on the pH is the success of the goldfish. So the dependent variable is a way that you measure the outcome. Maybe number of days or number of hours, if you want to be really specific, that the fish is alive. If you could quantitatively measure something else about the fish's health, that could be very revealing as well. So the dependent variable is how the independent variable affected the organism. If we were to graph it, here's your y-axis, here's your x-axis, here's our 0. Typically, the independent variable is going to be down here. Here's your independent variable on the x-axis, and over here on y is your dependent. Just like with math, if you said, oh, y equals 2x, x is the independent variable, and literally y depends on whatever you do with x. So if I put a 2 in for x, of course y is going to be 4. If I put a 3 in for x, of course y is going to be 6. So this really relates to math, of course. Science and math are quite related. If we were talking about pH here, well, we would probably have to adjust this 0, but we could have, 7 be here. Right here could be, 8. And if 6 were here, well, that's what I meant by adjusting where the 0 is. But my point is that you would have different plotted points for how successful these fish are. And that corresponds to the dependent variable. Here's the number of days or hours or months, whatever it might be, that the fish is alive and successful in that particular environment. And you would have an indication based on these plotted points. Oh, it turns out that, maybe it's 7.2. That could be the answer in terms of like, hey, that's the pH where the fish live the longest. And you notice that the way this is set up, it can be replicated. It can be duplicated by another party. The scientific method is never, well, I got this answer, and if you were there, you would have seen it too. I swear that's how it happened. It's going to be hard for you to replicate it, but I saw it. The whole point of this is that you want it to be as objective as possible, measurable, and able to be duplicated. Here's another example of the scientific method. This is actually based on a lab that I did in college. The question is, what color or wavelength of light is most effective at providing energy for photosynthesis? So which particular color of light in the spectrum of light is going to be best at making the plant healthy, making it able to make sugar for all of its metabolic needs? So the hypothesis, based on your observations of plants, you might think, oh, well, plants are green. I'm thinking they're going to respond best to green light. And I know that there are definitely a lot of plant parts in nature that are not green, but the majority of plants have a lot of green in them, and that's because of chlorophyll. But let's say our hypothesis is, hey, I think green light, that's going to be the most effective. Well, we got to test it now. So the control, like we said before, mimics the natural environment. The control would be white light. light from the sun. And that's natural. That definitely is mimicking the natural environment. That's what plants evolved in, white light, that they're used to absorbing all the different wavelengths or all the different colors of light. The experimental groups would be the individual colors by themselves being shined directly on these different plants. Red, orange, yellow, green, blue, indigo, violet. Roy G. Biv. And you see that down here with this illustration. The Roy is over here to the G and the Biv. And you can see the wavelengths of light in terms of the nanometers. Light is electromagnetic radiation, and the wavelength from one wave to the next, this is the actual measurement that's been calculated for these different colors. And you can see that the wavelength is much smaller when we get towards the violet. This down here is zoomed in on just this particular section. But there you go. There's the Roy G. Biv. And ideally, yeah, we would have every part of the spectrum represented in the experimental groups. The independent variable, what we are manipulating based on the question, is the color of light. That's the only thing we want to be different about those different boxes. And the reason why I say boxes is, I'll illustrate this for you on the next page. You want to have the plant isolated from all the light in nature, right? You don't want to have a bulb with green light shining on the plant while all the other light in this room or outside is shining on it. You want that plant to be isolated and just under that particular wavelength of light. Otherwise, you're not going to know whether or not it was the green light that had that effect on the plant. And of course, the dependent variable, you can measure it various ways. If this is a plant that naturally keeps growing vertically, the height of the plant could be directly related to how successful it is in terms of making sugar and being successful as an organism. If it's something like moss, which doesn't grow tall and just grows, sort of horizontally out, then height of the plant would not be as effective in giving us a good measurable result. The way that we did it in college was we actually measured amylase levels, and amylase is an enzyme that has to do with the breakdown of sugars. So the amount of amylase that the plant was producing was directly related to its metabolic health in terms of the amount of sugars it was making. It's only going to produce a lot of amylase if it is in turn producing a lot of glucose or sugar through photosynthesis. But height of the plant could be just as reliable in terms of giving you an answer. All right, an illustration of how this works. Here we go. All right, here is the control, and here's the experimental groups. And in the experimental groups, you're going to have red, you're going to have yellow, which I know is harder to see, but that's what it says. I don't have an orange marker. It's okay. You're going to have green. I'll represent most of the colors here, but not all of them. Blue, and of course, violet. And here are the plants all in their separate little boxes. The control, let's say here's our potted plant. Let's draw a little tulip. That's an easy one to do. So hey, here's the control. Here's the light in the box. Ding! That's supposed to be white light. Okay, since it's a white background, it's hard to represent white light. But whatever kind of bulb or lamp that you use in this, you would want to make sure that the wattage, the power of the bulb, the amount of light that's being emanated from it is the same across the board. You don't want these bulbs to be stronger than this one. The only thing you want to vary is that independent variable, and that's just the color of light. So of course, you've got your red light here, you've got your yellow light and so on. I won't draw them all quite yet, but we want to make sure all the other factors are the same. The size of the pot, the soil, the amount of water you're giving them. The area of the room you put them in should be the same, because on one side of the room where you're doing this experiment, maybe the temperature is 1 degree difference. Maybe light from the windows is heating up the boxes and you're not even aware of it. So you want to try to control all those other variables as much as possible. And that's going to make your answer more reliable. You want to know just how the light is influencing the results. So after we did this, it turned out that with the green light, that plant, I'm just going to do a sad face. That plant did not do very well, because as it turns out, the plant we used, it wasn't a tomato plant, but it was a species similar to that. It was a very green plant, and when you shine green light on it, it reflects green light. The reason why a green plant looks green to you is it's reflecting that wavelength off of those chlorophyll molecules in the cells, and if you just shine green light on it, it won't have really anything to absorb. So that plant did very bad. It died. With yellow and violet, it did okay. It did okay. So I'm going to give violet just a sort of a medium face, and yellow, hard to see, but there it is, medium face. They did okay. The total height over the few months that we did this was pretty good, or in our case, the amylase levels per little unit inside the leaves were decent. But the winners were definitely red. Red did very well, almost as good as the control, and blue did very well. And so, of course, we had to graph all of our results, be very specific quantitatively about how well they did relative to the control. And our results were not surprising to our professors, because it turns out that when you look at chlorophyll and its ability to absorb wavelengths of light, it's really good at absorbing reds and blues. Those are the two big ones. It does absorb, some from the yellow, some from the violet, but not nearly as much as red or blue. So these wavelengths of light, these colors of light, very effective at allowing the plants to absorb that light and have an energy source to make sugar. A little bit more on the scientific method. It's okay if you make a mistake. It's a learning process. And in that particular course that I took, where students were designing their own experiment and they were given a few months to do this, some of the groups actually had a big mess up, a big mistake, and they had to restart. But you know what? When they made that mistake, they learned from it and they didn't do it again. And that actually sometimes improved their setup the second time or the third time. Speaking of mistakes, I have a picture here with a little stamp commemorating Sir Alexander Fleming. Alexander Fleming made a mistake that led to a huge discovery. He was supposedly growing bacteria colonies on these petri dishes, these little plates. And he was exposing them to different conditions, etc. And he realized that on a plate, there was a whole region, and he definitely put bacteria on it, but there was a whole region that was devoid of bacteria. Nothing was growing there. And he's like, that's odd. So he took a sample from that region of the agar, the medium that the bacteria is supposed to be grown on, and he tested it, and it turns out a fungus, a mold, accidentally got on the plate and was inhibiting bacterial growth. So he didn't want that to happen, but it did happen, and he realized, hey, it turns out bacteria can't grow in the presence of that mold. And you know what? That mold is how we got penicillin. So that particular mold species, Penicillium is the genus name, we got penicillin from it, and that has saved millions of lives. A lot of people are allergic to these days, so you don't see it as much in emergency rooms or in treatment of bacterial infections, but nonetheless, this discovery that happened on accident really impacted the world in a positive way. So it's all a learning experience. These experiments need to have the ability to be duplicated time and time again, like I mentioned before. The scientific method is not about well, I did this and I saw it happen and you just had to be there. It's not about that. It has to be as objective as possible. Quantitative is preferable. Qualitative sometimes works, but you want to be able to duplicate this in another lab with other scientists over and over again. And if it can be duplicated time and time again with the same results, you're on your way to having a decent theory and maybe eventually a law regarding some kind of phenomenon in nature. Peer review. The whole point is when you're a working scientist, you're in a lab doing this, you want to be able to publish your results and have other people look at them and comment, make suggestions, learn from that experience, because everybody has their own kind of encyclopedia of knowledge in their head in terms of what they've studied and what they bring to the table. And scientists, it's supposed to be a team effort in terms of we're just trying to get to the answers in terms of how the world works, and we all can learn from each other and expand upon previous results. And adjustments may need to be made with new discoveries. As time goes on, there are new discoveries in terms of how cells work, in terms of what's going on in the brain. And if there's a new discovery that refutes a previous theory or refutes previous information that was gathered through the scientific method, that's okay. It's not like with certain religious beliefs, for instance. There are certain dogmas that can't be questioned. You just have to believe those things and have faith about those. But science is based on physical evidence. And if new physical evidence is refuting something from the past, it could be that the way it was discovered in the past was just not effectively done. They were missing something and maybe misinterpreting something. So new discoveries, they are welcome as long as we get objective empirical evidence that tells us about how the world works, tells us about how life works. Thanks for watching Educator.com.